Welcome to today's lecture. Today we were going to talk about immunoglobulin functions. Last section we have talked about the structure of the immunoglobulin. So as you see that the variable region of the immunoglobulin contains antigen binding site. So the first function of the immunoglobulin is specific binding antigens, where the variable region. It will cause the antigen recognition, neutralization, and agglutination. The first, antigen recognition. Because TCR and BCR share the similar structure, both of them have the antigen binding site, so they recognize the antigen in a similar way. So today we'll talk about them together. When the TCR and the BCR recognize the antigen by the variable region, so they will release a stimulatory signal for both of cells. Then these cells can be activated and then induce the adaptive immune response. This is the initial stage of the immune response, adaptive immune response. But here I would like to emphasize that TCR and BCR, they recognize in different mode. BCR recognize the antigen, that antigen is an intact antigen. That means it can recognize the antigen expressed on the surface of the pathogen. While TCR recognize the antigen need help by the antigen presenting cells. For example, dendritic cells is antigen presenting cells. When the dendritic cells encounter with the antigen, they will take up the antigen and process the antigen into small pieces. Then this small pieces peptide will bind a molecule which is called MHC molecule. Then they form a complex. Then they will express this complex on the surface for activation, for recognition of the TCR. So T cells only can recognize antigen presented by MHC molecule. So this is difference between the BCR and the TCR recognition. And we will talk about in detail of this step in the following sections. And next, neutralization. Neutralization refers to the antibody combined with antigen, and their binding could neutralize the virus and the toxins. For example, here is the virus. For example, it is a virus. And as you know, if the virus invades into our body, it cannot survive in extracellular fluid. It must invade into the target cell for its survival. And virus can express some molecules. Those molecules combine the receptor on the surface of the target cell. Then they can invade into the target cell and then cause the disease. But in the presence of the antibodies, those antibodies combined with the molecules expressed on the surface of the virus. So their binding will block the invasion of the virus into the target cell. There was no invasion. There was no damage. So this is called neutralization of the immunoglobulin. And neutralization is also useful to prevent the effect of the toxins produced by the bacteria. And next one is agglutination. Actually, agglutination is a phenomenon when the antibody binds the granular antigens in vitro. So agglutinate can be visualized. This agglutinate is antigen-antibody complex. So this red blood cells are very good granular antigens. Our red blood cells can express some antigens. For example, if the red blood cells express A antigen, so this blood type is A blood type. If the cells express B antigen, that is B blood type. If the cells can both express A and B antigens, that blood type is AB blood type. If they express nothing, that is O blood type. So if we can use the antibody, we can identify the blood type in vitro. For example, this is the antibody against the A antigen, and this is the antibody against the B antigen. So these antibodies mixed with the red blood cells. If we observe the agglutination, so we can identify the blood type, ABO blood type. And this is called agglutination. And agglutination is also can identify the species of bacteria. Those functions is mediated by the variable regions. And what's the functions of the constant region? As you know, the constant region combined with some molecules and immune cells, so they also can mediate some functions. The first one is they can activate complement. And complement is a component in the serum. When the antibody and the antigen, they form the complex. This complex can activate complement. And finally, they will form a membrane attack complex on the membrane of the pathogens. They form a pore on the membrane of the pathogen. Finally, they will lead the lysis of the bacteria. This is called activation complement, and we will discuss this in detail in the section of complement. Next, some immune cells can express Fc receptors, and they can bind with Fc fragment of the immunoglobulin to mediate opsonization, ADCC, and degranulation. So first look at the opsonization. Opsonization refers to the antibody promote phagocytosis of the antigen by phagocytes by signaling through the Fc receptors on these cells. You can see this picture, and the right one is present a bacterium. And the bacterium can bind with their specific antibodies to form a complex. And our macrophage is an important innate immune cells. They also can express the Fc receptor on the surface of the cell. Then the Fc receptor bind Fc region of the immunoglobulin, so they can pick up the antigen-antibody complex. Then they form a vesicle. This vesicle will then fuse with a lysosome. Finally, the lysosome, as you know, contains a lot of the enzymes, so those enzymes can cleave the parasite. This process is called opsonization. And this antibody, mainly IgG can work as an opsonin to mediate opsonization. Next one is ADCC. Here, ADCC means antibody-dependent cell-mediated cytotoxicity. It refers to the link of the antibody bound to the target cell with Fc receptors on a number of cell types, including the NK cells. They can direct the cytotoxic activities of the effector cells against the target cells. Look at this picture. That is a target cells. For example, it is a virus-infected cell. And those virus-infected cells can bind with the antibodies. And then the NK cells can express Fc gamma receptor. This receptor can bind IgG, Fc portion. So after their binding, they will transmit stimulatory signal for NK cells. then NK cells can be activated. NK cells is a very important innate immune cells. It contains intracellular granules. Those granules contain some mediators, for example, porphyrin and granzymes. Once they are activated, those mediators will be released, and then they will leading to the apoptosis of the target cells. So the process is similar to opsonization, but for ADCC, the target cells are mainly tumor cells and virus infected cells. Next one, degranulation. Degranulation refers to that, as you see that in our body we have mast cells and basophils. Mast cells usually exist in the tissues, and basophils in our blood circulation. And those cells are similar. They perform the similar functions. They have intracellular granules, and within the granules they have some inflammatory mediators, such as the histamine. And once they are activated, those mediators can be released out of the cells. This process is called degranulation. But how can these cells can be activated? Mast cells and basophils express a unique receptor, which is called Fc epsilon receptor. Fc epsilon receptor. And this receptor combined with IgE antibody. So after their binding, when the mast cells or basophil exposed to some antigens, those antigens can cross-link by the adjacent Fc receptor. So they will transmit a stimulatory signal for mast cells. So the mast cells can be activated and degranulate. This process is called degranulation. And finally, some class of the immunoglobulin can cross the epicellular layers. Transport proteins that bind to the Fc regions of the antibody carry the particular isotopes across the epicellular barriers. For example, IgG is the unique immunoglobulin which can transport to the placenta. So mother can produce the IgG which can transport it to the fetus circulation, provide the protection for the fetus. And next one is IgA, secreted IgA. Secreted IgA is very important for the mucosal immune system. IgA secreted plasma cells usually located on below the basement of the epicellular layer. And those IgA should be transported to the external surface of the epicellular layer to perform their functions. And they need to be transported across the epicellular layers. The epithelial cells express polyimmunoglobulin receptors. This receptor combined with IgA, and then they will internalize the IgA and then transport it to the surface of the epithelial cells. Once releasing the IgA, polyimmunoglobulin receptor should be cleaved, and small part of the receptor still on the surface of the immunoglobulin IgA. This part of the receptor is called secretory component. It's on the surface of the IgA to form a secretory IgA in the mucosal system. So secreted IgA is very important for the mucosal system. That's all for today. Thank you for your attention.